Hello and welcome to part 3 of our lecture on microbial genetics. Here we will be talking about different types of genetic mutations and when they occur, and a few repair mechanisms that may be used to repair those mutations. So in genetic mutations, we are looking at changes that occur to the genetic code found within an organism. There are two basic groups of mutations. We have what are called spontaneous mutations, which occur during normal cell processes, and induced mutations that occur due to some type of environmental factors, such as a chemical or maybe UV light, irradiation, something that is taken into the cell from the environment. So spontaneous mutations are usually due to cell processes such as maybe a mistake during DNA replication. Also, transposons can insert into different parts of a chromosome. Transposons are pieces of DNA that move from one region of a chromosome to another, and they can sometimes insert themselves and cause mutations as well. Things in bacteria, we could have changes during conjugation or transformation, those sorts of processes. But they're normal cell processes. They are not processes. They are not induced by introduction to some kind of mutagenic compound. Within the spontaneous mutations, we have two basic categories that occur. We have base substitutions and what we call frameshift mutations that occur due to additions or deletions. And then, of course, down here at the third one, we also have transposons. Base substitutions, we're going to look at the three different types of mutations that occur due to base substitutions within the genome. And then we will look at the types of mutations that occur due to frame shifts because of an addition or a deletion of a base within a gene. So induced mutations, we have three basic types of chemical mutagens that occur, chemicals that modify the actual bases themselves. We also have some compounds, such as some drugs, can actually act as base analogs. There are some antibiotics that act as a form of a base, and they're used to induce mutations. Remember, we're talking about bacteria here. So they're actually used to induce mutations so that those bacteria will accumulate enough mutations to finally not be able to survive. Then we have compounds that are referred to as intercalating agents. These are chemical compounds that will insert the actual chemical molecule itself into the DNA between base pairs to push them apart and start breaking up. Transposons are sometimes introduced. These are pieces of DNA that will randomly insert themselves into other forms of DNA, and transposons are usually introduced through conjugation or something like that. And then radiation. Radiation includes our UV light and X-rays. In our spontaneous mutations, these are the ones we're going to concentrate on. And here we did talk about induced mutations, and you should review those different forms. But in spontaneous mutations, these are occurring in normal cell processes, and the majority of your questions on your exams and such are going to be regarding spontaneous mutations. Now, these mutations occur in spontaneous mutations. We can have what's called a base substitution, and that's what we're going to look at first. So in our base substitutions, we alter or change one base for another. So we have the substitution of one base for another. Up here in the top right, we have, they're showing a mutation here. The normal codon that's being encoded for here is CAT, and that codon results in addition of a histidine amino acid at this location within the primary sequence of a protein, right? So this is the protein here, and this is the gene that encodes for that protein. If I change this A to a C, this is now a different codon. And that different codon encodes for a different amino acid, so that now my protein, the primary sequence of my protein, is no longer the same. And if proline has drastically different chemical characteristics from histidine, this could possibly result in some type of change to the folding process of the protein, and now that protein will not function properly because its shape is incorrect. An example of a missense, this is known as a missense mutation. An example of a missense mutation that results in a severe disease is sickle cell anemia. Sickle cell anemia is a single base substitution in the human genome that causes hemoglobin to not fold properly. And without folding properly, it cannot hold on to enough iron atoms to absorb oxygen. So it does not transmit oxygen well, and it also is not the correct shape. So the entire cell itself has a different shape. So this is referred to as sickle cell anemia. That's in eukaryotes. A nonsense mutation is another form of base substitution, but this base substitution, instead of resulting in a new codon and a new amino acid, it happens to encode for a stop codon. If it encodes for a stop codon, then you can see here my protein is cut short. I don't continue translating past that. So if I hit a stop codon, my ribosome, when it hits a stop codon, falls apart and no longer translates. So my protein that should have been 100 base pairs long is now maybe only 20 base pairs long. And that's an incorrect protein. It's not a non-functional protein. Now, the last form of base substitution that I don't have an example of here is a silent. And in a silent mutation, we are substituting one base for another, but in doing so, we have no change in the actual codon or in the actual amino acid that codon is encoding for. So let's go back to our codon table, and I'll show you what I mean on a silent. Go back here. So on the left here, we are looking at the codon, our codon wheel or table. And if I have a codon that is, let's say, UGC. UGC is a codon that encodes for, let's see here, UGC encodes for cysteine, or CYS. But if I instead create a codon that's UGU, this codon is a mutation. I've mutated from a C to probably an A in the genome, but this results in a mutation to a completely different codon. It's no longer UGC, it's now UGU. But if I look at UGU, it still encodes for cysteine. Remember, this is why we have the genetic code is called degenerate, because we have more than one codon can encode for the same amino acid. And these mutations, these missense and nonsense mutations and such, the hope is something like, let's say, for example, valine. Valine, I can change my third position to any of the 4 bases, and I still get a valine amino acid at that position in my protein. This allows for flexibility in that genetic code. We need that flexibility so that these mutations do not always result in non-functional proteins, and that more often than not, those mutations will result in the same protein. We just have a different gene sequence. So now let's look at frameshift mutations. Frameshift mutations can occur either through spontaneous mutations or induced mutations, and they are the result of insertions or deletions. Both an insertion or a deletion, both of these mechanisms will result in a change in the frameshift. So in the reading frame. So remember, the reading frame is set, right? We have we have the 5' end of our mRNA molecule, and there are some nucleotides there, right? We're looking at an mRNA. And then we have the Shine-Dalgarno sequence. And right after that Shine-Dalgarno sequence, remember the Shine-Dalgarno sequence is a flag that says the very next AUG. So there could be a couple of nucleotides here, but until we hit AUG, it now knows, the ribosome now knows that the very next AUG is where we start. And it does this in order to set the reading frame. Once we hit that start codon, we know every 3 bases. And our ribosome is going to move 3 bases at a time. In an insertion, if I insert a base right here, right, and I put a 4 here, this is now going to change my reading frame, which will ultimately result in a completely different protein. So in this case here, we have an insertion, it looks like. And so something was inserted here, and this moved from, I've shifted my reading frame. So my reading frame was normally, again, my CATs, but I have an insertion here of a T. You see this T here? And so now this has changed my reading frame. And my reading frame is no longer CAT. It's no longer going to result in this amino acid sequence here. It's going to result in this one, which is a completely different protein. These guys are going to, this primary sequence here, these amino acids are going to interact differently, which is going to cause secondary folding to be different, tertiary, quaternary, and my protein does not fold properly. And this occurs whether I insert a base or delete a base. Either way, I'm going to end up moving that reading frame by 1 or more bases, and it just throws off everything after that. When these mutations occur, we have to repair them. So in DNA repair, we have 2 basic types, right? We have types that repair spontaneous mutations and those that are used in induced mutations. And these are the different classes of them. So in these repair mechanisms that are used in spontaneous mutations, we have proofreading. In proofreading repair, DNA polymerases, usually during DNA replication, are going to proofread as the DNA is actually synthesizing. So it's moving across our single strand of DNA, and it's kind of rotating around, and DNA polymerase 3 is putting those bases on, and if it makes an incorrect match, this changes the shape of the DNA molecule, and polymerase 3 will recognize that and tear off that nucleotide and then replace it with the correct one. So these mistakes, when mistakes are made by polymerase, it most of the time will catch those mistakes. And it'll bring in enzymes it'll say, hey, wait a minute, guys, come over here. We need to take this off. I made a mistake. I put the wrong base there. And then once it's repaired, polymerase can then move forward and continue functioning. In a mismatch repair, this is where a mismatch has occurred and was not caught during proofreading. So if we don't catch it during proofreading, we're then going to, we do have like polymerase 2, which is a proofreading enzyme. That's one of its primary functions. It will move along a newly replicated piece of DNA. It is not synthesizing anything new. It is just reading the DNA and looking for abnormal bases or abnormal matches. It will then bring in enzymes and use something called excision repair to cut out. You can see here on this diagram in step 3, we've cut out where the actual mismatch was. This was a G bound to a T, and that should not have happened. So it cuts out not only the missing bases here, but just to be sure, it'll cut 2 or 3 bases on either side of it. Cuts those out and then brings in DNA polymerase 1 to replace those bases, and then DNA ligase to come in and repair the sugar phosphate backbone. The last form here on this page for spontaneous mutations is repair altered base. In a repair altered base, there's a specific enzyme called DNA glycosylase. DNA glycosylase will remove a base that has been damaged. So in this case, it's not that there was a mistake made during base pairing, but perhaps during replication or maybe during gene expression, transcription, something like that. Somehow, somewhere along the way, a base got damaged. And so there's a missing atom, or there's a bond that's not made correctly. It's not hydrogen bonding properly. The base is getting old or used and fragile. In this case, DNA glycosylase is an enzyme that will look for and find bases that are not completely intact, remove them, and replace them. For induced mutations, the 3 mechanisms that are often used are photoreactivation, In photoreactivation, we have to repair what are called UV dimers. UV light has a really, in bacteria, there's a specific wavelength of light called a germicidal wavelength that has the ability to penetrate across cell membranes and cell walls. And it will penetrate across and cause thymines to form these dimers. So instead of our thymines just being base paired to the A's, they are base paired to the A's. They are also base paired to each other. And so these thymine dimers, when these thymine dimers are created, you can see right here, it causes this lump-like structure. In photoreactivation, light energy is used to break this bond so that my T's are no longer in a thymine dimer formation. Excision repair is a mechanism that sees distortions in genetic material. Now here, the distortion is due to thymine dimers, but this can be due to other properties as well. And excision repair is a process that can also be used in spontaneous mutations. SOS repair is used when there's just massive amounts of damage. Oftentimes, long-term UV exposure can cause multiple mutations over multiple areas of the chromosome. And in SOS repair, there's a whole lot of damage going on. So it's an SOS polymerase. It's a specific enzyme that runs around and looks for large sections. And so it's excision repair where instead of 4 or 5 bases, we might have 30, 20, or 30 bases that need to be repaired, and that's going to require a different enzyme. So that's known as an SOS polymerase. Remember plasmids. Plasmids are those small circular pieces of DNA found in bacteria. There are eukaryotic organisms that have them, but we're concerned mostly about microorganisms. Over here on the left, these are plasmids, right? So we can see how small these plasmids are in comparison to the actual chromosome of the E. coli cell itself. These plasmids are self-replicating. They copy themselves, and so they can be transferred. So the cell does not have to be dividing for a plasmid to replicate. Plasmids are the common form of genetic material that are transferred during conjugation, and plasmids contain far fewer genes than chromosomes do. The genes they do contain tend to be what are called factors, such as resistance factors for antibiotic and disinfectant resistance, different forms of metabolism, something called dissimilation, genes for toxins, and genes for toxins against other bacteria. These are referred to as bacteriocins. Transposons are an interesting genetic anomaly. These are small segments of DNA that have the ability to move from one region to another, and they look a lot like a gene. So here I have a transposon that is a very large piece of DNA here. These yellow bands are indicating specific sequences that are transposase specific. Transposase is an enzyme. That's this enzyme right here. And transposase recognizes where these yellow bands are. It recognizes these two DNA sequences. It will attach to the transposon and pull these two yellow sequences together to grab the transposon itself and pull the transposon out, and then the two blue regions will be welded together. Once the transposase has this copy of the transposon, it will find some region within the genome to insert that transposon. And when it does, it oftentimes will do it in the middle of a gene or a regulatory region. And if we insert a bunch of genetic material, this is an insertion mutation. Now, this is a normal cell process. Eukaryotic cells have lots of transposons, and transposition is this process. Transposition is often associated with development of cancer. But in bacterial cells, transposons are often associated. This is how we go from an F positive cell to an HFR when we start moving pieces of genetic material from the bacterial chromosome to a bacterial plasmid. And then that plasmid is transferred to another bacterial cell through conjugation. When transposons, this process again is called transposition. When transposition occurs, the recipient cell of the transposition process is referred to as a recombinant. So that would be a process of recombination. So here's just a couple of quick review questions that you want to maybe write down and be able to answer these questions. You would see these topics on your exam. What's the difference between spontaneous and induced mutations? What are the different forms of these types of mutations? Are there any of these mutation mechanisms that might occur in both spontaneous and induced? Be sure to be able to define the different repair mechanisms, and when presented with a type of DNA damage, be able to determine what type of repair mechanism would be used. Be able to define transposons and have a general concept of how those transposons work and how they're associated with horizontal gene transfer, directly or indirectly. And how transposons are related to DNA mutations. What type of DNA mutation do they induce, and what type of repair mechanisms might be used to repair that type of mutation. So that's the last lecture, part 3 or 4, I'm not sure which, I think it's part 3. This is the last lecture in the microbial genetics, chapters 7 and 8. The final lectures will be on chapter 9 in biotechnology. See you soon.